Today I'm going to introduce my student who's the first speaker, Gali Bai. And Gali graduated with BS in agronomy from China Agricultural University, Beijing, in the summer 2018. After graduation, she joined my program for master's study in maths. Particularly, she's interested in big data analysis because she sees the tons of data, genomic data, certainly the VAS data, or phenotypic data, all many data is needed to be analyzed using more powerful bioinformatics tools. Gali is actually my third bioinformatics student. Students like working on big data analysis using bioinformatics tools. And she joined in 2018, and maybe before 18, that means this is her second year. Research show actually more like progress her research in this area. I like her. Okay, Gali, your turn.
Thanks, Dr. Hongbin, for your introduction. Today I'm going to talk about my research topic on genome-wide association analysis of agronomic traits and their heterosis in maize. Maize is one of the most important crops around the world, for it is used in the food, fuel, and feed. Though the yield of the maize has increased over 8-fold over the past decades, the increasing demand of the maize and the fluctuating climate changes have led to the need for a genomic improvement for the maize breeding. As we all know, most traits related to maize yield traits are quantitative traits, which means they are accumulated actions of multiple loci, multiple genes, and are higher interacted to the environment. So in this condition, a genome-wide association analysis of maize can be a powerful tool to maize breeding. My research objectives can be in these 3 steps. First, we genotype the maize inbreed lines to identify SNP markers. And then we collect the phenotypic data. And then we integrate the genotypic and phenotypic data. And finally, we find the markers related to the traits. My plant materials are from the very popular inbreed lines which was widely used in GWAS. Among 302 core lines, 226 maize inbreed lines were selected. They have 7 subpopulations: NSS non-stick stock, SS stiff stock, TSS tropical and subtropical, popcorn, sweet corn, and maize. The field trial was conducted in 3 years from 2017 to 2019 and in 2 locations, on College Station and Lubbock. Only the 2018 field was conducted in 2 locations, and for the 2017 and 2019 were only conducted on College Station. All the inbreed lines were grown in 3 years, and also other lines were planted in 2 replications and in a randomized complete block design. We collected 5 years per line per replication for phenotypic data. We first phenotyped these 7 traits for the yield traits. Most of the traits show a normal distribution despite the grain weight and year weight, which shows a little bit skewed distribution. In 2018, we also collected the heterosis phenotypic data. From the picture, we can see that the hybrid lines in the back have a significant strong phenotype compared with the inbreed lines. Heterosis of grain yield, 500 kernel weight, and plant height were collected in 227 lines. Then I used BLUP, fast linear unbiased prediction, to shrink the data from multi-year, multi-location to the mean. I assign the line, location, and year as random effect for this linear model. Heterosis then were calculated based on this formula. We can see that among the 7 yield traits, 3 of them shows a higher heterosis. 3 of them shows a higher heritability. And the heritability are ranged from 0.48 to 0.87. Also, a correlation was conducted. We can see that green weight has a high association with year weight, and also green weight has a high positive association with kernel length and ear diameter. And then the genotypic part was conducted in high-performance clusters. I first downloaded the sequence data from NCBI, which were sequenced on the Illumina HiSeq 2000 platform. And then I used an SRA toolkit to transfer them to FASTA file. Because those data are not sequenced by our lab, so I first checked the quality score of those FASTA data. And those data show a high thread, which are average above 32. And then BWA aligner was used to do the mapping. And then samples were used to do the variant calling process. After genotyping 263 inbreed lines in the reference genome B73, about 7 million SNP markers were collected. Those under the strict filtering threshold, like minus alleles, frequency 0.08, missing data 0.5, and quality score 30. There are also a large amount of SNP markers remain. So I plot the SNP markers to see how much SNPs I can screen for the next step. And the plot shows that there are still a lot of SNPs remain, even though the minus allele frequency were conducted from 0.08 to 0.5, and missing data were selected from 0 to 0.5. Also, I used the structure to analyze the subpopulation of my inbreed lines. The structure results shows that there are 3 subpopulations, which are consistent with another research, which used SSR marker to find the subpopulation of 302 inbreed lines. We can see that there are 2 major inbreed lines, which are non-stiff stock and tropical and subtropical in the left picture, which correspond to the green and red part of my result. During this whole process, I find that BLUP is not showing a good predictive ability when we use the other effect as random effect. So next step, I may choose BLUP to train the model with genotypes and fit the genotypic effect as fixed effect, and then compare the results to the BLUP. And also, I still have a lot of SNP markers which are over 7 million. And I think it is too much for GWAS analysis, and maybe there are a lot of false negative results. So I will choose a more strict threshold to filter those data. And for the next step, I may use this genotypic data and phenotypic data to do the GWAS analysis. And then for the heterosis, I will use the B73 and inbreed lines to find the F1 heterosis genotypic data and do the GWAS. That's all my process until yesterday, and I hope I will have the opportunity to show my next steps in the future. Thanks to my committee members and Texas AgriLab Research and Corn Production for their help in the field analysis. Thank you, and welcome to any questions.
What are the mixed lines?
I think mixed lines are the seeds are mixed from the SS, NSS, and TSS.
Oh, I believe that's integrated more like three major subpopulations: tropical, subtropical, SS, NSS. The three major subpopulations for the corn. And I believe somehow there's an integration during plant breeding, whatever. So those lines is not belongs to any subpopulation. That's why a cluster into another one called a mixed. When you see the scores, actually more like somewhere 0.4, 0.5 something. It's not like a 0.9 above or close to 0. So that's when you use the structure software, you will see that.
Have you tried to use BLUPs on individual year data to see if there's better alignment with a particular year?
I thought to do that, but BLUPs shrink the data to the mean. So I think if I use the individual year and shrink individual year and then shrink to the whole data, it will generate too much errors. So I
just
didn't.
year by itself in comparison to see how big is the environmental effect is on the trade.
I will do that. Yes.
You mentioned that you had a large VCF file and you think you're getting false positives because of that. Have you considered tagging your SNPs to reduce the SNP panel?
Tagging the SNPs to reduce SNP panel. What do you mean by tagging the SNPs?
So a lot of the SNPs are probably going to be in perfect linkage disequilibrium, which means that they're going to be 100% predictive of other SNP. So you can just remove redundant SNPs and that'll increase your power without losing resolution.
Yeah, when I run the structure program, I filtered the SNP markers based on the linkage disequilibrium. Yes. But I don't know if I should filter all of them because I didn't see people do that.
Well, they're 100% predictive of one another, then there's no reason in keeping those that are redundant because they're redundant.
Thank you.
I put some comments. Actually, the missing data filling, she used per low stringency, which is 0.5. Suppose they put a 0.2, which keep anything the marker with missing data fewer than 20%. So she used 50%. After cut, maybe the SMT number will drop, maybe 1 million and a half million. That's something we expected. Maybe there's 2 million or 1 million somewhere. So the minus allele frequency, that's another major important for GWAS character. That's when she used 0.08, which actually that's make 0.12 or 0.15. So that minimize the false positive locus identified by GWAS. Another thing that we're doing is here. Actually, that's the data she collected. There's this population, there are a lot of phenotype data. There are 60 traits people did nationwide. They published them. There's no GWAS analysis. And most important, there are also heterosis. They did multiple years, multiple locations. They published that there's no GWAS analysis. That's why she worked on this project. She planned to grant you the summer. That's why she shouldn't do the presentation fall, but she planned to grant you the summer. So that's why she have no choice. She have to do this semester.
And also, I think the reason why I got that much of SNPs markers because when I do the variant calling process, I put all the 264 BAM files to the VCF and make it to call the variant, so it may produce much more SNPs or yeah, right, than the other method.
Yeah, so I don't disagree with any of that. The point that I was making, if a SNP is under 100% linkage with another SNP, you don't need to keep that with SNP because it just wastes computational power.
Yeah.
It's just more work for you.
Thank you.
Other questions? You know, you 681 students, you've got to ask questions. You've got to ask 3 questions. It's a good opportunity for you. Okay, let's thank our speakers.